Let's get some water.
We should start. I mean, there are people missing, but they will
That's
come.
all right. That's fine. Yeah.
Let's start first lecture of our fourth course by Paul Rainey. So it's a pleasure to have you here. Thank you for coming. I just want to announce, not for you actually, but for the other people not here, that we should try to start at 9:30. So. Because we can pass on the message.
Well, they've figured out I'm a biologist, and that's not too much turning up for. That's quite okay.
Okay, so please.
Okay, thank you. So good morning, welcome. I hope you had a nice evening, you rested well. I'm sorry for my voice. I've gone from winter to summer to winter to summer, and I'll go back to winter in the last 3 weeks. And each time I seem to have picked up a cold that, or maybe I've been spreading them as I've gone. The evolution of individuality, then. To me, it's one of the most fascinating, challenging, and complex problems in biology. It really gets to the heart of the evolution of biological complexity. It's been, for a long time, in the domain almost exclusively of philosophers of biology who've made important contributions in terms of trying to work out what matters. But at the same time, I guess a lack of mechanistic thinking has led to many of the issues being very murky and opaque. There's definitely been the need for experiments, and a number of labs have begun to delve into some of these central issues. And at least in the case of our work, this has led to some insights that were surprising, which has led to the development of theory that will take what I present today, that comes from the realms of experiment, and attempt to generalize it. And at least from my perspective, what has emerged, and will be clearer, I hope, not tomorrow, but on Thursday on my second lecture, is that what seems so shrouded in mystery, almost to the point where you would want to invoke a divine entity as explanatory power, is in fact quite simple. The evolution of individuality is tied up with life's hierarchical structure. And I will use this set of Matryoshka dolls, John, Paul, Window George, and the mysterious fifth beetle, a number of times to capture the sense of life's hierarchical structure. So the evolution of individuality is very much about why life is as it is. And why life, I would conjecture, on another planet will have exactly the same kind of hierarchical structure. There are 4 seminars. This topic could go on for months, but I would like to take the opportunity to speak about another topic when it comes to lecture 4. So the first 3 lectures then today will go through an introduction, scope, define what matters, and the focus will be experimental investigation of the evolution of individuality. The second lecture then will take what we've learned from the experiment and simplify it using a very abstract mathematical model that attempts, and I think reveals, that some of the specific issues that you will see today are in fact generalizable. And the concept that I will present is referred to as ecological scaffolding. I will touch upon it today. It's a way of understanding the relationship between organisms and their environment that has profound effects for the evolution of individuality. So this concept of ecological scaffolding then, I will take on in lecture 3 to look at the evolution of heredity, one important component of individuality. So here's an email exchange with Luca. He wrote to me at the beginning of last year saying, would I come along? And I wrote back to him saying, thank you, it would be wonderful to come along, but you do realize, and he does know, that I'm not a mathematical biologist. And he says, sure, we know you're not a mathematical biologist. It would be fun to see your interactions with them. And I said, you mean like meat to lions? And he said, no, more like taming lions. So, this is a nice exchange, but it also draws attention or reminds me to say something about,
Sorry to interrupt. Yeah. Could you just wear the medallion and turn it on? I forgot. Sorry about that.
Okay. To say about the relationship, of course, between theory and experiment, it's not lions and tamers. In fact, they're, of course, complementary, very complementary. I probably don't need to tell you that in biology, theory is important in many regards. Often it's used, as you saw yesterday, Luca, Josh, that it can be a way of abstracting and formulating complex processes. It can be used to construct null models against which we might compare experiments. But it can also take experimental observations, as I will show you on Thursday, and generalize and abstract. So there is always this wonderful interplay between experiment and theory. So hopefully you have a good understanding here. Oops. All right. Some relationship between pushing the button. Let's try an experiment. Well, okay. Let's try this. So today then, the first lecture, individuality. Of course, you're dying to know what I mean by individuality. I'm going to tell you. We'll look at that in the context of life's hierarchical structure. Then I'll spend a little time trying to explain what I think out of all of this stuff really needs to be explained. Okay, thank you. And then we'll move, for at least a half of the time available, to consider the evolutionary transition in individuality as we go from individual cells to multicellular organisms. And that's an experimental aside. So individuality. We are individuals, but I mean something a little more. I mean individuality in a Darwinian sense, which indeed we are. But what makes us Darwinian individuals? First of all, a Darwinian individual participates in the process of evolution by natural selection. And you may go, so what? Well, where does one start? Would you go, so what? Do you understand why that is so important? Let's imagine evolution without this process of natural selection. Could you conceive of that? What would it mean? It would mean that evolution depends on chance and chance alone. Now, it's a common mistake, actually, to think that evolution is driven by chance alone. It's not. A lot of people that have difficulty with coming to grips with what evolution is about will say that how can chance alone generate complex life? And you would respond, but evolution is not based on random chance alone. Natural selection, although it depends on variation being generated randomly, brings about a systematic change in populations of entities. In fact, let me put a very succinct definition of evolution by natural selection, which I think owes to Richard Dawkins. Evolution by natural selection is the non-random, the non-random survival of randomly generated variation. So variation upon which selection acts is generated randomly, but selection biases the success of that variation. It brings about a systematic change in the structure of populations. In the absence of selection, we wouldn't have life. Random things can be great, chance is a fine thing, but if there is no process, when we understand selection, that amplifies what works, there is simply no possibility of life getting going. So these Darwinian properties then that I'm going to explain to you are essential for this process of evolution by natural selection to operate. Again, evolution by natural selection is a strange beast. Even the philosophers that have thought deeply about it struggle to put their finger on what it really is. It's not a force, although it is sometimes referred to as a force. It is a process that unfolds provided three conditions are met. These are the conditions of individuality. First of all, there needs to be variation among entities within a population. These types differ one to another, and that variation needs to affect their reproductive success. The second requirement or property is reproduction. Here we see that the one in the center has left 2 offspring, the one on the side 1, the one on the left hand none. Their variation has manifested as differential output in reproductive success. So one property variation, the second property reproduction, and the third property for selection to work is that offspring resemble to some extent the parental types. Now, provided entities within a population manifest these 3 properties, they will participate in the process of evolution by natural selection. That's it. They will. Completely substrate neutral too. Computer algorithms that manifest these properties will similarly participate in the process of evolution by natural selection, provided these 3 properties are met. So a Darwinian individual is an individual within a Darwinian population defined by the fact that the entities manifest these 3 properties. Often they're referred to as a single term, that there is within a Darwinian population heritable variance and fitness. It captures the essence again that there is variation, reproduction, and heredity, but I'd like to spell these properties out: variation, reproduction, and heredity. Now they're not actually separate to one another, they're intimately linked, as will become apparent as we move on. So the evolution of individuality, about which I wish to speak, is really about the evolution of these Darwinian properties: variation, reproduction, and heredity. Now again, if you pause for a minute, that may seem an odd thing for me to say. And the reason why you might think it's odd is that if we look at life, all life manifests these properties. We all vary one to another. We reproduce. Our offspring resemble us. That's true of fungi, of bacteria, of trees, of shrubs. Doesn't matter. It's therefore easy to look at extant life forms, see these properties, and say, well, they are simply features of life that don't need explanation. But they do need explanation. The evolution of individuality is about the evolution of these most fundamental features of life. Life's hierarchical structure then. So I've emphasized John here, who is standing in for a multicellular organism. He is a member of a population of multicellular organisms, such as we are members of, is a Darwinian individual. He participates, like we do, in the process of evolution by natural selection. But we could delve down inside of the body of John, or me, or you, and we would find that these same Darwinian properties manifest at different levels of cellular and genetic organization. So let's do that. So we rip me apart, and perhaps the first thing that we see are tissues and organs. Let's take the heart. To what extent does the heart participate in the process of evolution by natural selection in its own right? And how would you even assess that? Well, the simple way to do it is simply, do hearts leave heart-level offspring? Is there some replicate replicative capacity at the level of hearts? Well, the answer is no. So these levels do not reflect tissues or organs. But if we go down inside tissues and organs, we see that they're comprised of cells. If we apply the same questioning, do cells have some capacity for replication? The answer is clearly yes. So cells manifest these same Darwinian properties that we manifest. It'd be slightly disturbing to think about that. We can go on a little further. Down inside of the cells, within a eukaryotic cell, of course, you will find organelles, such as mitochondria in us, and in plants, mitochondria and chloroplasts. And those organelles, as you perhaps know, are derived from once free-living bacteria. They even come with their own little genome. Those organelles also reproduce, replicate. They participate to some extent in the process of evolution by natural selection. We could go further down into the nucleus. We find chromosomes. Clearly chromosomes have those Darwinian properties. We can go inside of chromosomes, if you like, to the level of genes. And you might think that's a bit odd. Genes just replicate as part of chromosomes, but take surface genetic elements, transposons and the like, and we clearly see that genes also can replicate themselves, sometimes independently of the rules of Mendelian genetics, and therefore manifest these Darwinian properties. Now, we could, in principle, go on back. It becomes more abstract and more difficult to recognize discrete stages, but right on back to what I conceive of as the origins of life from non-life, the point at which we get self-replicating chemistries. And those chemistries, to begin the process of life's origin, must have some capacity to participate in this process of evolution by natural selection. That means, at the most fundamental, some capacity for replication. We could, in fact, go above the level of the multicellular organism without controversy. We could go to eusocial insect colonies, for example, that have these same Darwinian properties. And there are some blurry areas in between. Some would like to think about culture as having these sorts of properties, or the microbiome as having some of these Darwinian properties. These are issues that need to be sorted out, and they have important implications. Because if selection acts at all of these different levels, then the product of this is adaptation at all of these different levels. And the implications of that are profound. So there's a sense here, too, of time, that as life's history has played out, lower-level self-replicating entities have become subsumed within higher-level self-replicating structures. So single cells, for example, that have given rise to multicellular organisms. Multicellular organisms are self-replicating entities comprised of the cells that were once free-living. So again, what this brings us to is a sense of transitions, levels of selection. In fact, I'll go on to another slide in a moment where we'll capture that. Here we are, which gets us into some really murky area. And I hope by the end of Thursday that what will appear perhaps murky now, perhaps less murky after the experiment, will be crystal clear once we move to Thursday's lectures. But it has been typical to think about life's hierarchical structure in terms of, first of all, major evolutionary transitions in individuality. Wonderful book, more than 20 years old, by John Maynard Smith and Seth Mowry, that recognized that life was organized like this, that there had been a series of major evolutionary transitions where genes became part of chromosomes, where cells became part of multicellular organisms, major evolutionary transitions. Each of these different hierarchical levels is said to be a level at which selection operates. Well, that's not too difficult to understand because each of these levels manifests some of those Darwinian properties. I mentioned this a moment ago, a sense that life's complexity has evolved by a series of transitions in which lower level entities have been subsumed within higher order self-replicating structures. When this happens, selection is said to have shifted level. Selection operated at the level of cells and has shifted to operate at the level of multicellular organism. Now that has caused me a lot of confusion How does selection shift? Selection can't shift. Selection isn't some force that decides, today I'm going to operate at the level of multicellular organisms. Selection can only work when those Darwinian properties, variation, reproduction, and heredity, manifest to some extent. So as I say here, simply, selection doesn't choose. Selection is a process that unfolds when certain conditions are met. So we're thinking about cells to multicellular organisms. Selection is seeing cells because they have those Darwinian properties. Cells begin to form groups. But selection can't work on those groups unless the groups now manifest those Darwinian properties. And we will look exactly at that in a moment. So selection needs handles. By handles I mean these Darwinian properties upon which it can draw. Now again, there's been a lot of talk, and I'm going to talk about it too, to the sense that when selection does shift, the fitness of the lower level decouples in some way from that of the higher level. I won't even say any more at this stage. I'll show you that it does happen from an experiment. I will try and articulate why that has happened, but on Thursday, I hope I will show you that actually there's no great mystery there. Now, something I haven't mentioned. Selection at different levels is typically in conflict. So we function, of course, our function is utterly dependent on the cells that make us up. And those cells, for us to function and remain healthy, must play by the rules of development that are laid down by the multicellular organism. And that requires that cells stop growing when instructed, undergo apoptosis, cell death, when instructed. And clearly that is not to the short-term advantage of the individual cell, but it's very much to the advantage of us as a multicellular organism. Our cells need to work, if you like, for the good of the collective. Clear evidence that individuality manifests at the level of our cells is cancer. So cancer arises typically when mutations occur in cells. That means they no longer play by the rules of development or obey the instructions of development. So whereas development said, stop dividing, this mutation now says, I'm not listening to that instruction, and the cell grows unchecked. This is, in the short term, highly advantageous to that individual cell, even if it causes the death of the multicellular organism. Selection is blind, typically, to future consequences. So selection acting at these different levels brings about an awful lot of complexity, a lot of tension. From a mathematical point of view, thinking about how to describe this tension is challenging. It involves building models of nested Darwinian populations where timescales in particular, multiple timescales, over which selection operates in different ways, is crucial. And here's a question posed by Samir Akasha, a philosopher of biology who's made very important contributions to thinking about major evolutionary transitions. He poses this particular question. I actually think now it's not a good question to pose, but it's not a bad one to get us going, because it really makes you go, what on earth is going on? So, recognizing this tension between what's good for the cell and what's good for the multicellular organism, and thinking about cells becoming multicellular organisms, here's a question. What would cause the lower level entity, cells here, to give up their own right for autonomous replication, and in so doing become parts of corporate bodies where they're slaves to the collective? So, putting that differently, we haven't got multicellular life around, they're just single cells. They're participating in the process of evolution by natural selection. They're autonomous. If we look at multicellular organisms, where cells are no longer free to evolve in their own right, they are part of multicellular organisms, what happened in the past that tricked, if you like, cells to give up this autonomy and become part of a multicellular organism? Again, I hope you will see that it has a very straightforward answer. It's not perhaps the most productive way to ask the question, but nonetheless, there is a sense of mystery here. So major evolutionary transitions then. Lower level entities becoming subsumed within higher level self-replicating structures. That's the hallmark. David Queller pointed out two kinds of transitions. And I think this is very useful because it points to two different kinds of problem that need to be solved. So David pointed out that there are transitions of an egalitarian type and transitions of a fraternal type. Transitions of an egalitarian type involve the coming together of two free-living entities. The evolution of the eukaryotic cell from two once free-living bacteria is an example of an egalitarian transition. There are many ideas about how this happened, but the prevailing view is one cell type either consumed or invaded another. Let's come up to, whoops, okay, sorry, out of sync with my own slide, that's not good. So egalitarian transitions coming together of two different sets of entities. The transition to the eukaryotic cell is such an example. The fraternal transition is what we're going to talk about today, that happens from within, if you like, involves a single cell that gives rise to a group. Aha, we seem to have hit the same problem. Never mind. So two kinds of problems are posed. For these egalitarian transitions, we have two entities that already have these Darwinian properties. There's no such thing as niceness in biology. We like to think that things cooperate and, they're kind. I think for the most part, that doesn't work. Even if these two different cell types found it beneficial to live in each other's presence, for example, some cross-feeding, it will be unstable. Selection will always favor types that take advantage of the other. It stands to fall apart. So what needs to be solved is the alignment of reproductive fates. That requires the evolution of heredity, and that's what we'll talk about in the third lecture. For these fraternal transitions that come from within, heredity is not a problem, so to speak, because the individual cell that starts the group gives rise to daughter cells that are identical. The problem that is posed here, though, is the problem of reproduction. How does this group reproduce? Okay. So here, the problem was solved by one cell subsuming another, either invading or subsuming. But when that happened, the reproductive fates became almost perfectly aligned. With regard to reproduction, we've got an individual cell that was a mutant. It produces offspring cells that stick together for some reason. It's evolutionarily beneficial. This group grows in size. And if nothing happens, it just keeps getting bigger and bigger and bigger and bigger, and there's no reproduction. So it needs to find some way of giving rise to two or more. That could involve fragmentation, but if we look at the multicellular world, I do know of one organism that will reproduce by a kind of binary fission, but for the most part, even organisms that don't have a soma-germ distinction reproduce via a single cell bottleneck, and you will see from experiments why that is so important. But this is not a trivial thing to achieve. For the growth of the body, reproduction through a single cell bottleneck, growth of the body. In many organisms, of course, there's a dedicated germline sequestered early in evolution. Okay, let's do that, yeah. Thank you. Yeah. Okay, it's good. So it's a dedicated germline. Now, this is clearly evolved, but the evolution of reproduction here poses many challenges. Integral, too, to this process of going from one single cell to a collective of cells is development. The evolution of development is intimately tied with the evolution of reproduction. So these two kinds of transitions, coming together as separate entities or happening from within, as in the case of multicellular organisms, pose two problems in terms of explaining the evolution of individuality, and you will recognize that heredity and reproduction are different aspects of these components of individuality. So turning now particularly to multicellular organisms, a prelude to the experiments. You'll see this cartoon a number of times. It's my way of trying to simplify things in my own mind. The first stage I think of as the evolution of simple groups. Individual cells here that are participating in the process of evolution by natural selection give rise to some simple undifferentiated group. Living in close proximity to one's neighbors is always costly. Intense competition for resources, dealing with the waste products of your neighbors additionally imposes a cost. Such a group will not remain viable unless there is some collective benefit to the group that offsets those costs. Assuming there is some benefit to the collective, then theory tells us, an experiment reveals that theory is correct, that we will see the evolution of defecting types, often they're referred to as cheats. I try to limit this kind of language as much as possible. Types that don't contribute to the collective good, but nonetheless take advantage of what's provided. We could think of these as members of a society. We are all paying our taxes. We reap some benefits, metros, libraries, university, from contributing at some individual cost. Because there is some collective benefit, it will pay for individuals not to pay taxes. Now, of course, if most individuals decide not to pay taxes, society falls down. So we have developmental programs in the forms of laws and policing systems to prevent that from overrunning the collective good. But just to draw an analogy there. So the first stage is the evolution of simple undifferentiated groups, but the problem of defecting types is a real problem. If they become common, they stand to erode that emerging collective. The next stage, and remember the title of this lecture, The Evolution of Individuality, is about exactly that. To make that clear again, here we have cells where those cells are members of Darwinian populations. This stage here results in collectives that have those exact same Darwinian properties. Once we have collectives that beget collectives, in just the same way that cells beget cells, then we have a Darwinian machine. This configuration of life will participate in the process of evolution by natural selection, and the outcome will be some form of multicellular, complex multicellular life. Its exact form will depend upon many factors, particularly the environment. But that's the easy step. This is the difficult one. This is about the evolution of these properties at this higher level. The fact that they exist at the level of cells does not allow them to automatically transfer up to the level of collectives, which has often been assumed. So again, just a brief prelude here, touching upon things we've already mentioned. But the evolution of multicellularity, one of those major evolutionary transitions, is all about the evolution of these Darwinian properties of variation, reproduction, and heredity that ensure that collectives participate in the process of evolution by natural selection. They need to be rediscovered at this transition. They don't automatically transfer. I mentioned at the outset, it is commonly assumed that these properties, these Darwinian properties, are given features of life, and we don't need to explain them, but we most certainly do. So how do we explain their evolution once we recognize that reproduction and heredity, and actually the bedmate variation, are derived traits? Well, you look at something like reproduction in multicellular organisms. It's a complex process. It's natural, therefore, to look at that and say, well, the cause of that is natural selection, because that's all we can resort to. But of course, we can't do that, because that would be invoking the thing that we want to explain, reproduction, as the cause of its own evolution, and that simply will not do. And that, if we take nothing for granted, and one could take things for granted, but I want to take nothing for granted, takes us to a very interesting place. which is recognizing that we need to explain the evolution of these Darwinian properties, but from entities that don't have those properties, non-Darwinian, and therefore by non-Darwinian means. Now that suggests that chance is the only way out of this predicament, and that's not going to work either. So what I'm going to argue, we don't really, well, I've already made this point. We don't need to. All I wanted to make a point of again is that once you have a cell that produces offspring cells that stick to itself, it grows and grows and grows and grows and grows. The individual cells are reproducing. They have the Darwinian properties. But this collective of cells is just going to get bigger and bigger and bigger. And unless it can break apart in some way, there is no possibility of a Darwinian process at the level of the collective. It would be sufficient just for it to break apart, although there are problems with that happening, but that would get things going. So how do you make progress? The idea that I will present more fully on Thursday is that these Darwinian properties can be scaffolded by the environment. The experiment I'm going to show you now is a good example of this actually working. But I won't go into the more abstract ideas around scaffolding at this point, but simply that these Darwinian properties can come from outside. So now to some experimental studies where we're going to deal with this problem of how reproduction of collectives emerges. So for a long time we've worked with bacteria. They're Pseudomonas, they're common bacteria. You will eat them when you eat salad. They're common soil plant colonizing bacteria. They're not pathogens. This particular isolate, Pseudomonas fluorescens, SPW25, came from a sugar beet plant at the University of Oxford's farm in Wytham. When you grow it on an agar plate, many of you will perhaps have seen a bacterium on an agar plate. It produces a smooth colony. If you put it into a test tube and leave it there, it will consume the resources. Competition becomes intense, particularly for oxygen. Many mutations are arising as population size, 6 mils of media with around 5 times 10 to the 9 cells per mil, so a very high population size. Standard mutation rates, it means in there's a huge amount of variation that's being produced, very strong selection. What happens in this test tube is in fact a miniature adaptive radiation. Many different types emerge that specialize at different niches within this file. That story we're not going to go into, but I'm going to draw attention to what we refer to as a wrinkly spreader. You can see it's wrinkly and it spreads. It's a descriptor of its colony morphology. That's not so vitally important, but what is important is that this mutant type here, which evolves from here by numerous point mutations, simple, one mutation is sufficient, but there are many roots, forms a simple undifferentiated group. Now, I'll show you how this works in a minute. Use some cartoons. Cells, although they're rod-shaped cells in reality, I'm going to represent the individual cells as red spheres. And these wrinkly spreader sticky types have Velcro on the outside. It enables them to stick. So here's a test tube. Media goes in goes the ancestral type. It grows, replicates by binary fission. About 1 in 10 to the 6, 1 in a million cells will be a mutant to the wrinkly spreader type. So in a population size of around 10 to the 10, there are many wrinkly spreader mutations. After 24 hours growth, the doubling time with this organism about 40 minutes. So these mutants arise. They overproduce a glue, a cellulosic polymer. They don't separate properly at cell division time, and they're able to form this mat at the air-liquid interface. Now this really is an example of individual cells contributing to a collective action that reaps a benefit at the level of the collective. It's costly for these cells to overproduce cellulose. It's a polymer of glucose. It's better to metabolize glucose than excrete it or secrete it. So these cells grow slower than these cells here. However, these bacteria need oxygen. Growth, metabolism in the broth phase eliminates oxygen within about 12 hours. The only place there is oxygen is at the air-liquid interface. This mutation, causing overproduction of glue, allows the cells to remain attached to one another. They spread across the surface. They're not buoyant. They spread across the surface though and attach themselves to the glass vial. The collective of cells within this mat has plenty of access to oxygen. So access to oxygen, a consequence of these cells building this mat, offsets the individual cost of producing the glues. It explains their increase in frequency, despite the fact that individual cells of the sticky type grow more slowly. That mat becomes a substantive structure. Again, the cells are reaping all the benefits of oxygen. As we would expect, this sets the scene for the evolution of types that don't produce the glue. These grow effectively like tumors. You can see them with your eye on the surface of the mat. These cells don't produce glue. They arise by mutation. They enjoy all the benefits of being in the mat, but they pay none of the costs. They weaken the structure of the mat, and the mat collapses. So here we've seen the emergence of these simple undifferentiated groups, but also faced the reality of these cheating types. We saw this just a moment ago. These three stages. So you get from here to here very readily. However, here is a problem. And the problem of defecting, cheating types has long been recognized. Rick Michaud, one who argues that the evolution of this complex multicellular life form is totally dependent on the evolution of means of controlling cheating types. And while I'm with them, we maintain our integrity by all sorts of self-policing systems that punish cells that don't obey, eliminate them. But those mechanisms are complex. How do we see the evolution of something so complex when we don't even have entities that participate in this process? So this really is a problem here at this stage. What we know we need is reproduction. I want you to keep this in mind. This is an example of scaffolding and Darwinian properties. I will explain this much more on Thursday. But what you see here inspired the experiments that I'm going to describe in a moment. What you see here, even if not self-evident, is you have to imagine a pond. And in this pond are reeds extending out of the surface. And these reeds provide opportunity for microbial mats to form. So I showed you a test tube within which you saw the mat. You could ditch the test tube and just have a reed, and the mat will form around the reed. So what we've got here are 6 reeds. There is an enormous amount of variation among these wrinkly spreader types, these sticky mat forming types, indicated here by different color. We've got variation at the level of these collectives by virtue of this environment. Discreteness comes from the environment. Now let's imagine that this yellow mat fails, stick falls in the water, and it falls down. There's a vacant niche there, which is available for occupation. Let's consider the possibility that via some means, red cells are able to migrate, disperse, and form a mat around this reed. Well, what you have unfolding here is a Darwinian process that's taking place, or stands to take place, at the level of collectives. These collectives here have these Darwinian properties. They're externally imposed, they're not endogenous features of the organisms. We get discrete variation. We've got something that, well, it's dispersal, but it looks a lot like reproduction. We could draw a phylogenetic tree in which we represent these 6 different colors. The yellow comes to an end, the red branches. There really is the possibility of a Darwinian process unfolding. So keep this in mind. This is what we're going to see, but done in experiments. Now, if you follow this, then attention shifts to the mode of dispersal, which is kind of a proxy for reproduction. Is that just fragmentation? That would kind of do, but fragmentation will never get multicellular life very far. The main reason is that within the just take ourselves, within the lifetime of the body, as we get older, we're more and more likely for mutations to arise that will give rise to cancer, various other degenerative diseases of aging. If we just split ourselves in half when we reproduced, all of those conflicts would be passed to our offspring. So fragmentation will always be problematic. The problem of cheating types won't go away. It just doesn't work in the long term. So we could think about non-arbitrary fragmentation. Maybe all of the cells fragment into single cells and they all go off, as some multicellular organisms do. We could think about life cycles involving multiple states. We could imagine that the mat is kind of like a soma. It performs an ecological role, getting oxygen, and maybe it produces propagules, like a germline, that can then give rise to the soma-like state again. But of course, if you think of that, you think, it's just too complex. How could that possibly come about? And in the face of these wonderful ideas, we recognize that defecting types remain a problem. So here is a very specific detail of this experiment, which I won't generalize, but it does provide a route by which a complex life cycle can emerge almost for free. So we've seen this a number of times now, cells to a simple undifferentiated group, the problem of the cheating type has to be solved before we get to this stage here. Well, what if we turn this problem here to a solution? Could we turn it to a solution? Well, the answer is yes, given particular ecological circumstances. So here are two reeds in a pond, two different genotypes. There's the emergence of the inevitable defecting types. And in our experiments, these don't produce the glue, they're motile and they're free to move away. So it moves off into the pond. The green one disappears through some disturbance, but this defecting type aids dispersal and allows the reformation of a type that is a direct descendant of this mat over here. So the idea that these defectors then could in fact be a solution to a bigger problem. The bigger problem being, how do groups beget groups? Well, maybe these propagules here, these defectors that have the properties of propagules, might in fact allow this process to unfold. So we're going to look at that now in an experiment. And the regime I will talk to you first about is the one that we refer to as the cheat-embracing regime. It doesn't seem to be the obvious thing you would do, but it does embrace the problem of the cheats and turns it into a solution. What this shows here is a life cycle that begins with a single sticky cell that forms the mat. We see these asocial defecting cells emerge, which are a problem, but we embrace them as part of the life cycle. They become phase 2, and for the life cycle to complete, it needs to go back to produce a sticky cell. This is how I would represent it. In a while, I will do the experiment that you might have been voting for, that we refer to as the cheat purging regime. It's exactly the same, except when the cheats inevitably arise, we get rid of them. We purge them. And so we go from one cell to a big mat and back through a single sticky cell. Here, single sticky cell, big mat, but now we transfer this through the second phase before we get back to a sticky cell. Here represented like this. Now, I know many of you are not biologists here, but a biologist would have problems with what I've just put forward, in that what it is requiring is for this life cycle, going from sticky cells to non-sticky cells and back to sticky cells, to start as a consequence, or to be driven, initially at least, by mutation. No developmental program in biology is driven by mutation. The idea here was perhaps there's enough evolutionary flexibility underpinning the transition from sticky to non-sticky cells and back again to allow selection to begin to work on a developmental program, which involves forming a mat, a dispersing cell, forming a mat, to the point where it might even become an endogenous feature of the evolving organism. And I will show you here, work from a little while ago, that is exactly right. We see the evolution of a simple genetic switch a simple developmental program. Work that there simply isn't time to talk about here, we've seen that go much further, to the point where these transitions are now controlled in a truly developmental mechanism regulated by the availability of oxygen. What I'm showing you here is work from many years, actually, that's been parallel to this, that is focused on the different mutational routes to overactivate this glue. When this is overactivated, we have our wrinkly spreader. What you see here are three signal transduction pathways, the outer membrane, the inner membrane. This is what we refer to as the WISP pathway. These depict proteins, particular regulatory components, negative, positive. All three of these encode digranulate cyclases, DGCs. When they are overactivated, cells make too much cyclic di-GMP. This causes overproduction of the glues. Mutations arise in these pathways. There's three possible routes to overproduce the mat-forming type, or to produce the mat-forming type. Here's another set of 14 routes involving digranulate cyclases that can also be used by evolution. All of this to say that there is a remarkable capacity for evolution to work on. The genetic architecture underpinning the transition between the sticky state and the non-sticky state is highly pliable. There is hope that we might get off the ground given this kind of work. Two heroines, Katrin Hammerschmidt and Caroline Rose, who handled, I hate to think, it's almost cruel, but many tens of thousands of these microcosms during the course of 5 years, learning how to do these experiments. Daniel Rexon, who's now with me in Pleuren, and Philippe, who's now in Toulouse. The experiment now continues much more advanced at the Max Planck Institute for Evolutionary Biology. There's only time to talk about the first contribution here. Here's the life cycle. So you saw that pond. Remember the picture of the pond, the reeds, the mats. Well, here we ditch the reeds, we use test tubes instead. We take a single sticky cell. In it goes into a fresh test tube, forms a mat. This is what we call phase 1. It lasts 6 days. At the end of the 6-day period, we take that test tube, we vortex it, we plate. And on the plate, we can distinguish between the sticky cells, because they have a wrinkled morphology that make the mat, and the non-sticky cells that stand to be the propagules of the next generation. We collect the non-sticky cells. These are the second stage of the life cycle. And then we add them to a fresh microcosm. And at this point, these cells, during a 3-day period, must give rise, by mutation to begin with, to cell types that form this mat once again. At the end of the 3-day period, we check to see if that's correct, take a single sticky cell, and the cycle starts again. So a 2-phase cycle, a 6-day phase, a 3-day phase, during which this life cycle needs to play out. This is not easy for the cells to achieve. It requires, to begin with, again, mutation. Should the cells fail to produce the next stage in the life cycle, then we'd consider that lineage to have gone extinct. Clearly, it doesn't fulfill its life cycle. Additionally, the life cycle can fail and an organism go extinct if there is no mat at the end of the 6-day period. This would be the equivalent of an organism dying before the age of reproduction. We would simply say you're dead. So we run this experiment. This is the number of life cycle generations here. We started off with 120 lines. And you can see that after generation 6, all have gone extinct. And they go extinct for different reasons. We don't need to go into that. The point is, it doesn't look good, does it? But some lines did traverse 6 cycles. That required at least 12 mutations. Now, something that I learned from this experiment is the extraordinary difference in terms of the potency of selection operating on fecundity versus viability. Typically, it works on both. In the experiment I just described to you, these all went extinct because selection was solely on viability. We never allowed the possibility of fecundity. We said, can you replace yourself? And if you could, you kept going. And if you didn't, you simply came to an end. What we never did allow here is what you saw in the pond experiment. You saw in the pond experiment one mat collapse, and then another mat that was persistent at that time, it exported its success. So it produced, there were now two types. Under that scenario, we have selection on viability. You need to stay the course, you need to build a mat, but also fecundity. If you are a good mat former, then you get to produce, if somebody else fails, another mat. So here, 120 lines, everything went extinct within 6 generations. Selection was purely on viability. I've just said that. Let's now allow the possibility that selection works on fecundity as well. So now we take the same, or a different set of independent 120 lines, we organize them into 15 replicates of 8. This is the process here of lineage selection, where selection is working on both viability and fecundity. Our 8 microcosms. Here is one that failed because the mat collapsed. It's extinct. It's just like the mat forming off the reed. There is a vacant niche available. Any of these other types then can be chosen at random, it happens to be the next door neighbor, and allowed to export its success. So this one replaced itself, but thanks to this death event, it also got to export its success, have another crack. You know, I always like to point out at this stage that death is underrated. Evolution, thinking about evolution as death-birth processes that I very much do. To stay in the game, you just need to persist and hope that you're slightly better than somebody else that doesn't. And then there is this opportunity presented. Now, allowing selection to work like this, these 120 lines persisted on through 10 generations, at which point, in this early stage, we took this experiment apart. So the difference between selection and viability and fecundity is immense. Something else that I will particularly emphasize on Thursday. Well, first of all here, selection. What is required to succeed? Well, the primary thing is to be able to build this mat and then give rise to the propagule phase. It requires development in a very crude way. Selection is working on development, and we'll see it really does work on development. But the other thing that is happening here is, thanks to this life cycle, there is a second time scale that's emerged. We have 2 time scales. The first is the doubling time of individual cells, about 40 minutes. But the longer time scale is that of the doubling time of the mats. That takes 9 days. What's in the interest of individual cells is not necessarily in the interest of the nascent multicellular organism. Selection will favor individual cells that grow fast. They will win out within a microcosm. That may have been the case here. A very early arising cheating type that killed the mat. Selection over the long time scale sees that one eliminated. It's gone. Although selection favors fast growth within, over the longer time scale, that's not at all favored. So selection over the second time scale kicks in, and it's extremely potent. It holds those lineages with cancerous tendencies, if you like, to account. They're eliminated. And that's simply a consequence of the second time scale. And that's something I will really emphasize on Thursday. I like to gaze at this. And this is just a tiny snapshot of what goes on in these lineages. This is the fate of every microcosm in these experiments. What you see, every node is an extant type, either a wrinkly type, a smooth type, wrinkly type, smooth. These types are persisting. Where there is no line coming on out, it indicates there's been a death event. You can see early on there's massive death. You would think there's no possibility of going on, but some go on and are extremely successful. In looking at this, it's unusual to be able to see the evolutionary fate of every single individual, but you see it. Now you might look at this and you go, well, here's a very successful lineage. What would we look at as a predictor of its success? You might use fitness. So we might come back here, follow it back. Let's come to this point here. Let's compare its fitness at this point with its contemporaries. Well, here we have a contemporary. It gave rise, I think there was something like 6 or 7 birth events there. But as you see, most of them went extinct the next generation, one continued one more generation and then went extinct. Whereas this one here left just 3 offspring, and yet it gave rise to this lineage here. Time is short and I don't want to go on about this, but seeing this kind of process, this birth-death process, has really changed my view of the currency by which we should measure evolution and the predictors of future evolutionary success. I can talk to you later. There are mathematical representations of this now that are really illuminating. An important thing again is death. I indicated here that this was the most successful type because it left 6 or 7 offspring, whereas this one only left 3 at the same time point. But actually, it only got lucky. It happened to be chosen 7 times because there was a lot of death. The fact that it left 7 offspring was not actually a measure of its fitness at all. So there's a lot of chance that goes on here. And chance, again, is a fine thing. And chance over the second timescale allows many things to happen. I won't go through that in the interest of time because I'll touch on it tomorrow. But just to give you a sense, so Guillaume Doucier, a biologist that turned mathematician, just finishing his PhD in Paris with us, has done many things, and I'm going to talk about his work on the evolution of heredity on Thursday in the 3rd lecture. But he built a simulation here which of evolution under different situations. So here's our standard fitness landscape. You will be perhaps familiar with this representation, the idea of a fitness optima. Here is a standard serial transfer. This is like the Lenski experiment. Multiple populations, this is their evolutionary trajectories. They all rapidly go up the local peak here when they start from the outside. There's little chance that they move on here. It's exactly what you would expect. If you operate this process of lineage selection with a second time scale, these same populations now, and this is just showing the last, I think, a couple of 100 generations of wandering, you can see they wander all around. They explore a great deal of phenotypic space, and the reason for that is the second time scale. There's this rapid, selection on the short time scale favors fast growth. Over the long time scale of our experiment or the simulation, fast growth is not the deal. It's doing something else, like playing the life cycle. And typically growing fast and doing something as complicated as unraveling or allowing to play out a life cycle are at odds with one another. The second time scale ensures that fast growth is no longer the only game in town. In the case of our experiment, it's doing a life cycle, a developmental program. And that becomes possible through exploration of phenotypic space that is only possible when selection is not rewarding outright fast growth. There's timescales and the math behind the timescales. There's some wonderful challenges. So to finish up, and time I guess is nearly, let me just tell you what went on after 10 generations of selection. So our experimental design, which captures that pond experiment, saw Darwinian properties imposed on these evolving mats. What we would expect, therefore, is to see mat fitness increase. That at the end of the experiment, the mat-forming genotypes should be producing more mat-level offspring than they were at the beginning. And we can measure that by competition experiments, taking ancestral mats, derived mats, using opposing genetic markers, and we can compete them. 1-to-1 initial ratio, and then we can ask what's the ratio at the end of the experiment. So here we start off with 4 in a single rack, 4 of this, 4 of that, ancestor, derived. After one life cycle, we see that the derived type is now occupying 6 of the 8 available spaces. So we would say the derived type fitness has improved. We can also measure cell performance, and of course we're interested in knowing what's happened to the cells, because after all, mats are comprised of cells. And one can relatively easily measure growth rate, density, number of cells in the mat, all sorts of different measures. So what did we find? Well, we saw that selection sees the lineages, the mats, quite differently than it sees or saw the cells. Here's the data. Line, this is our nascent multicellular organisms, this is our collective. And here are our cells. We measure fitness through a competition assay, and what we see is that after 10 life cycle generations, the fitness of mats has gone up significantly. That means that selection saw the mats. We imposed these Darwinian properties, selection worked. That's perhaps not surprising. These cells, though, the individual cells, and it doesn't matter what measure of cell fitness we have, they've all gone down. I should have pointed out, if there'd been no change in fitness after 10 generations, the data point would be on the dotted line. Individual cells here have experienced around 2,000 generations of selection. And as many of you know from Rich Lenski's work, that when you propagate E. coli over time and you measure fitness, there's a trajectory that goes something like this. And even within 1,000 generations, there is a significant, I think around 5% increase in fitness. So 2,000 generations, and we've gone the opposite way. Now, that's a consequence of that second time scale. As evolution proceeds, the cells that are successful are those that align their reproductive fates with that of the collective. Fast-growing cells are selected against, typically because that will challenge the ability of cells to play this life cycle. So what we see here is evidence of decoupling. The fitness of the collective is no longer explicable in terms of the fitness of the individual cells. Individual cell fitness has gone up, whereas individual cell fitness has gone down, collective fitness has gone up. And when we get to the theory, I will show you it's very simple to understand exactly why this happens. It's entirely intuitive once you recognize the importance of this second time scale. Nonetheless, this is what philosophers have been talking about. It does make biological sense that during an evolutionary transition we should see the emergence of a new kind of organism that is not simply the product of the individual fitnesses of the individual cells. We're going to talk about second timescales a lot in the next lecture. I'm going to conclude by telling you what happened in the cheat purging regime, because that's our null model, if you like. But I also want to tell you what happened genetically that gave rise to this fitness increase. So although we can't explain the fitness of collectives based on the fitness of individual cells, we should definitely be able to explain why the collectives have got more, have improved, why the fitness of collectors has increased, and it should have a mechanistic basis in individual cells. This simply shows that to begin with, the life cycle was prone to failure. By the end of the experiment, just 10 life cycle generations, these best lineages were reproducibly doing this life cycle with minimal failure. There's some liberty taken here, but just to show you the essence of what happened, you saw these three pathways earlier on. What you're now going to see is a red dot that indicates a mutation. That mutation causes the wrinkled mat-forming type. The next mutation there shuts down the overproduction of this, and you get the sticky cell. And evolution bounces around through these different pathways, switching between the different types. These are all spontaneous mutations, nothing special going on here. One realizes extraordinary capacity for evolution to go back and use what it's previously used. This bar here, diguanylate cyclase, I've highlighted that because that's been left in a phosphorylated state, which is necessary for it to be active and make this component. Eventually what happens, is a mutation elsewhere, but before I show you that, this bar, this is the active site of the enzyme, GGEEF. And right in the glycine residues is a string of 7 Gs. Now, strings of nucleotides are prone to mutate at higher frequency by a process called slip-strand mispairing. But 7 Gs is not sufficient to do that. Once you've got 10 to 12 or more, then there will be an elevated mutation rate in that tract. That's not sufficient for any elevated mutation rate. However, a mutation in MARTS next arises. This is an enzyme involved in DNA and methyl-directed mismatch repair, which is a DNA repair process. When that is mutated, the mutation rate of the entire organism goes up. But, as we had not appreciated, tracks of 7 Gs suddenly start to slip. So some chance history leaves WISPR in an active state. By chance, there are 7 Gs here. Closely related Pseudomonas, protein sequence is always conserved here, but there's a lot of redundancy in these codons. You hardly ever find another that has 7 Gs in a row. But this one happens to have 7 Gs active, and along with this, we now have a simple genetic switch. The reading frame is destroyed by the addition of a G. The reading frame is repaired. Back and forward, a gain and loss of G residues. And it works brilliantly. This genotype swept actually even beyond its own rack. Backward and forward as a consequence of a switch that has appeared as a consequence of this selective process, and this is the primary cause of the improved fitness, at least for one of the lineages here. I'll leave that comment aside. The causative factor, how important is this two-phase life cycle? Remember the cheat embracing, that's what we've just looked at. Two-phase life cycle selection had to work on a developmental program. Cheat purging, of course, we got rid of the cheat. What happened? Well, here is the same data, run 10 generations as before. You can see the fitness of the line has gone up, but unlike what you saw before, the fitness of the cells has gone up. There's nothing particularly interesting that's happened here. Through the selective regime, individual cell fitness has gone up, and that simply explains the improved fitness of collectives. No sense of decoupling. We explain completely the fitness of the collective based on the fitness of individual cells. Okay, there's a paper on the archive that looks at a different level of complexity here, but I'm not going to talk about that, other than to say, even when you've got a two-phase life cycle, something I'm going to say a little bit more and wrap up in a moment, it doesn't guarantee that you will move the fitness of groups up. Here we've got fitness of groups, and on the x-axis we've got fitnesses of cells. In our experiment, the one I've just explained to you, selection has pulled the fitness of the groups up and at the expense of individual cell fitness. There's really been the emergence of a new kind of organism. If we take that two-phase life cycle and allow mixing of the propagules, selection now sees that propagule phase much more intensely, and we pull selection along this route, and it favors the individual cells. We no longer see that decoupling. I can talk about it, or you can see it on the archive. So some concluding thoughts then. So life, I started out by pointing is hierarchically structured. These Matryoshka dolls representing it owes much to these major evolutionary transitions where lower level entities become are encompassed in higher level self-replicating structures. Each of these transitions requires a discovery, the evolution of these Darwinian properties. These properties can be externally imposed, and I'm going to elaborate much more on this. They can be conferred by the environment. The pond reed, you just think about this, those groups didn't know that they were multicellular organisms or anything. This particular ecological structure that simply provided a spatial discreteness in the environment and a means of dispersal is all that was necessary for a Darwinian process to unfold. It gets us out of this difficult situation, is highly relevant to thinking about many things, including the transition from non-life to the first chemical replicators. What I find fascinating here, and if you look at the transition to multicellular life, it seems to be an immense challenge to explain, as I mentioned to begin with. And if I told you that a life cycle replete with development could emerge in a short space of time, you would simply say rubbish. And I think you would, I would be sympathetic to that. But what we see in this one experiment is that there are a lot of things that seem to come together. What we see is the emergence of a life cycle, which simply disperses into a new habitat. That gives a second time scale to an evolutionary process. There's a birth-death process that now happens over this longer time scale, which is defined by this life cycle. We see a soma-like structure, the mat. It's an evolutionary dead end in and on its own, just like our soma. We see the soma-like structure perform an ecological role, acquisition of oxygen. We also see, and very effectively once we've got a developmental program, that those same cells that are performing an ecological role are also generating the seeds of the next generation of mats. There really is a reproductive division of labor going on. All of that has emerged from the simplest of ecological conditions and some frequency-dependent interactions among the different types. The two-phase life cycle seems to be tremendously important. What it is offering is selection. What it offers to selection is something that selection has not seen before. Just remember the cheap purging regime. One sticky cell became a lot of sticky cells and went back to one sticky cell. Selection is simply seeing sticky cells, and we see sticky cells improve their growth rate. The selection doesn't get to see anything new. The cheat embracing regime, though, selection can't just see the sticky cells, it can't just see the non-sticky cells. For it to persist, it needs to see both types and the interactions between them. That's something altogether new. Selection really has something to work with at that stage. And then finally, a comment here on life's hierarchical structure. And I posed that question from Samir Akasha, it just looks horribly complicated. Why would individual cells give up their own right for autonomous replication and come to replicate as part of a corporate body? Well, here you can see that there's nothing like that going on. All that's happened here is we had some benefit to collective living, acquisition of oxygen, some means of dispersing to a new environment when resources are depleted. A Darwinian process unfolds at this higher level. No one is being conned. It's really just ecology, eco-evolutionary dynamics unfolding under a particular scenario. This gives rise to life's hierarchical structure. And as I will show you on Thursday, in even simpler and more abstract terms, it really can happen extremely simply and with a minimal set of requirements. Let me just mention I've mentioned, so the theory, I'll mention other people tomorrow. In this early work was Ben Kerr at the University of Washington, Caroline Rose and Katrin Hammerschmidt, who've now well moved on, who were the major experimentalists behind this. So thank you for your attention, and let's take a break, and I look forward to coming back.
Well, actually, we are running a bit late, so maybe we should skip the break and go straight to questions, and then we take a big break before the next lecture, so if it's okay.
Absolutely, yeah.
Okay, so, yeah. Thank you.
I found very interesting what you were saying, this idea how we move from one hierarchy to the next has a lot to do with reproduction. For instance, how instead of one individual reproducing, now we have a group reproducing. And so I'm a bit curious about if our biology is paying attention also to the architectural constraints that you would require for a system to reproduce itself, right? For instance, because I think this is kind of a relevant question, how a collection of entities organized in such a way that it's able to reproduce itself, like what Francisco Varela called autopoiesis. But I'm not sure if this is something you're also trying to look at, or you just assume an entity that reproduces without concerns about the mechanism.
So we very much care about the mechanism. Here, in thinking about the evolution of multicellular life, reproduction in our experimental regime is involving going through a single cell bottleneck. So it is ensuring, I didn't really talk about this, that the next generation bears, essentially identity, is identical to the previous generation. So we have a very high level of heredity by virtue of being a fraternal transition where one cell gives rise to the group, and then we go back through one cell again. Under other scenarios where you don't go through a single cell bottleneck, and again that's something I'll talk about on Thursday, then of course the group that is reformed may not bear much resemblance to the earlier recurrence. I'm not sure if that's, but the mechanism of how reproduction takes place is critical.
Maybe an example to me is, for instance, the DNA, right? You have these double strands such that
Right.
it is able to do the reproduction, because this is kind of mathematics, this is called self-reference, it's a self-referential problem that is not always easy to handle. So I think in the case of the DNA, the architecture is two complementary systems, subsystems
Right.
that reproduce itself. There are more complex architecture that can do that I think Francisco Varela and Maturana were working about in autopoiesis. But I don't know if this is now like mainstream, or you're talking about this kind of how the molecules or the cells organizing this kind of structures, or is not?
Yes, so the problem will manifest differently depending upon the kinds of transitions, but the key thing is information transfer. With the transition to multicellular life, the way it stacks up, it's not so problematic. If you're going through a single cell bottleneck, then the next generation, provided it was founded from a majority type in the earlier recurrence, will take that information because it already has DNA in the chromosomes that take, it's got all of that stuff as given. So, but that mode of reproduction affects the degree of heredity. So that's almost certainly why multicellular life almost exclusively goes through a single cell bottleneck as part of its reproduction. It makes a fresh start, but of course it also poses the problem of development. If the multicellular body was performing a useful ecological role, and now you go through a single cell bottleneck, then how do you need to regenerate the next stage. And why it goes through a single cell bottleneck is not just because it's a fresh start, but basically that discretizes variation. And if you've got discrete variation, selection will see that configuration of life much more potently. So I think that's what, there's this interesting interplay between what selection sees, what selection amplifies. But we could talk more. I think the problems, the mechanism of reproduction and the consequences are really something that are of basic interest.
How do sporulating bacteria fit into this intellectual scheme? There's a particular one whose name I suddenly forget, Senior Moment, I think.
Bacillus, anyway.
Yeah, maybe it's that. In other words, when conditions are good, they grow and they form a colony. When conditions get rough, all of a sudden the colony undergoes collective motion, a stalk is formed.
Dictyostelium.
What is it?
Dictyostelium?
Dictyostelium, yes. They create spores which then get distributed by the wind, and it's a single cell bottleneck, blah, blah, blah. But everything is already, how shall I put it, the entire program of this two-stage life cycle is, so to say, within the genome of the stable organism itself.
How do they fit in? Well,
how does this fit in?
Well, we could talk a lot about Dicty afterwards. I don't think you should believe what you're told in the literature about Dicty. Dicty is a bit of it doesn't make a lot of sense, actually, from the perspective of it being a unit of selection that selection works on. The reason being, actually, if you look at it through the perspective of these Darwinian properties and you ask, to what extent does Dictyostelium manifest these Darwinian properties? It doesn't really. The reason being is that the stalk, so for much of Dicty's life, it exists as individual single cells. Those individual cells are doing their thing and they're participating in the process of evolution by natural selection. Periodically, but just how frequently, no one knows, they will form this fruit body, this slug, which then develops into fruit. Those slugs are typically chimeras. Multiple different genotypes are in that slug. So, yes, yes, this is the case. In the lab, that's not the case, but in wild, they're typically chimeras. Now, what that means is that you may have a brilliant slug, that's doing just its great things, but the next recurrence of that slug, if you trace the genealogy of the cells, it would bear very little resemblance to the parental type. So, how dicty fits into this is interesting. There are many life forms. What I would emphasize, what we are paradigmatic Darwinian machines. Life doesn't need to fit into that exact framework. What it will simply mean is, in the case of dicty, the question is, to what extent does a dicty cells, so where is the major impact on the evolution of dicty? Is it at the single cell stage, or is it when it's in a fruit body, or in the slug? If slugs really ever form relative to single cells, I would say most of the action is happening at the level of individual cells. But that then poses the question, what about this developmental complexity? But there is some interesting ecological models for that don't present it in terms of cooperation and conflict. And there's been some nice work recently published, actually, from a guy, Chris, I can't remember, in UCL, a nice pioneers paper that presents some lottery models. I could explain afterwards a little bit more. But dicty is, there are many organisms a bit like dicty that are in the grey area. They're not paradigmatic in terms of the manifestation of Darwinian properties at the collective level, but they're doing just fine.
So a simple question from a non-expert. How do you measure fitness? Is just counting cells or mass across generations?
That's effectively how it's done, exactly. We take the classic, Lucas had mentioned, in the Lenski experiment, so to measure cells, it's easiest. So you take, you've got your tube at the beginning, we call it our ancestral population, and what you can do with bacteria is you can store them in the minus 80 degrees freezer, so you've got the ancestor, you've got your great-great-great-great-great-grandparents stored. And then you've done some period of evolution, and we've got our derived type here. And what is nice with microbes is we can carry out a very simple measure of fitness, which is reproductive success of this type relative to that. And so what we can do is, this is also stored in the minus 80 freezer, so we can come along some later time point and say, what's happened during the course of X generations? So, using some neutral genetic markers, which we'll perhaps in here. So these ones might be, white, and these ones filled in color. It's neutral. So we can take them, put them into a single flask, equal ratio of ancestor to derived. We can count them on an agar plate, measure their ratio. Then we can grow them for 24 hours and resample and measure the ratio. So this should be, 1-to-1 ratio if we've done our experiment right. And after a period of evolution, we may find that there are more of these. And we can express this as a ratio of Malthusian parameters, and it gives you a very precise measure of fitness. However,
I noticed the error bars are not small.
No. Well, so Luca had mentioned frequency dependence yesterday. So fitness is rarely ever non-frequency dependent. And the moment fitness is frequency dependent, then this founding ratio is critical. If it's 1.01 to 1, that will affect the outcome. So in our case, fitness is massively frequency dependent. So yes. Yeah, it's not simple. Actually, it gives a sense, this is beautiful, you can do it, but for most instances, the conditions necessary are not really met, and no one really cares about it, because it's too complicated. Once you've got frequency-dependent fitness, you can measure it, but it's always dependent on that founding frequency. And yeah.
So Paul, you started with the Matryoshka dolls and this notion that cells can have replication, as does the full organism, but the heart and other organs can't. You can't take a heart and make another heart.
Well, you sort of can in the lab, right?
In the lab, but the heart on its own.
But not inside the body.
The heart on its own won't do it. And so in the course of this experiment, do you think you're going in the direction where you still have Darwinian replicators at all the scales, meaning if you had taken your cells, which then are not part of the mats, the sort of individual planktonic form, and you were to put them in a stirred environment that doesn't allow you to form mats, I presume they would still replicate. Yeah. And so I was wondering if you could speculate, do you see something like a heart forming at all, or what would you do to get there?
No, thank you for the question, because I should have pointed out to begin with, our work is using a bacterium. It's not a eukaryote. The major innovations in multicellularity have taken place in eukaryotes. part of the answer, I don't, so what we've done here with the bacterial system is to try and get at the early stages of transitions that should be, general features of the process. The extent to which we can get much beyond to getting, I think bacteria are particularly limited with their ability to evolve development where development involves differential phenotypic expression across space. Through time it's okay, but across space. And I think that's probably necessary to get anything that approximates a heart.
Yes, I mean, I was intentionally pushing it a little bit farther, but so my follow-up was really, if you've tried, when you showed those two, the fitness of the mat and versus the individual cells, one went up and one went down a little bit, but you could have run this whole selection regime, one with just in a mixed regime, so favoring the planktonic, and one in which you didn't require the planktonic phase by just selecting the mat and passaging mats.
Yes.
And I was just wondering, do you see that the developmental type does not have, in some sense, a pure mat fitness as high as one which doesn't have to go through the planktonic phase? If that makes sense. You showed two fitness, and I'm thinking about the ones on either side of it. One which doesn't have to go through a planktonic phase, a single cell phase to reproduce, because you can just select the mat and passage it to a new regime.
So that's, I guess, what we call our mat-to-mat treatment. And so there, we don't see the cells fitness going down. If it's just gone from mat to mat to mat to mat to mat, although it goes through a single cell bottleneck, then the fitness of cells has gone up. But I'm not sure.
Yeah, maybe I'll ask you later, slightly different version, but that's okay.
There are certainly, the conditions I haven't talked about at all, the selective conditions necessary to get this kind of working. It took us a long time to work it out. This process of lineage selection that we operate turns out to be very powerful, primarily because there's no mixing ever, but there is competition within groups. If you allow groups to mix during this phase, you don't see any of these effects, which is understandable, actually, from the point of view of these Darwinian properties. But we could talk at length about these. Yeah.
So this notion that selection can work on two time scales and see cells and lineages differently. So considering that, what is the relationship between these two different time scales of selection and the fitness landscapes?
Well, good question. Fitness landscapes. Tell me a little bit more. I mean, I'm with you with the time scales, and we'll definitely talk a lot more about time scales. But the connection with the fitness landscape, what are you thinking about?
So how selection acting on those two different time scales, how that would shape the fitness landscapes and how that might change the fitness landscapes?
Well, are you thinking particularly to that slide I showed?
Yes.
Well, what it's primarily doing there, and will do generally, so if you have, so the second time scale is typically working against selection favoring fast-growing types. So in the short term, fast-growing types will always be rewarded. In the Lenski type experiment, effectively fast-growing types always can be rewarded. If there's a second time scale, and actually how you implement a second time scale and so on is an interesting question, then you stand to have a situation where over that longer time scale, types that grow within a lineage fast will be eliminated. So selection ends up rewarding slower growth. And what that really means is that there is much more opportunity for drift and wandering and exploration of phenotypic space. That's not there if growing fast is the only game in town. To grow fast or to run fast, you need to fulfill a certain set of criteria. And that's running fast is going to trade off with ability to do many kinds of other things. So that analogy is not so far off. Basically, the second time scale, selection is off fast growth. There's more wandering to be done. And you can, the game is just about persisting then. If you persist and others fail, then there is another possibility to, a lot can happen. simply exploration of phenotypic space. Maybe you even, go down into something of a trough, but it comes with some phenotypic novelty. But that's fine because you continue to persist. So the short of it is it allows much more wandering and exploration of phenotypic space. So those simulations show around these populations basically just wandering. Whereas if you're just growing fast, they will always just go up the first local peak. In fact, in those experiments, most populations found their way to the global maxima, even though they started close to a local maxima. And it was because of the wandering. And the wandering is because fast growth is no longer being favored. And in the wild, fast growth is probably rarely ever favored.
Well, we have time for one more question, and then we are running really late.
Professor, so you stated that the second phase cycle was operating on a longer time scale, right? So I was sort of expecting a different measure of fitness for those colonies, perhaps somewhere where you would measure their ability to invade oil flasks, right? So isn't the competition experiment that you made in order to measure the fitness between the ancestral and the new type a short time scale measure of fitness?
No, because we are basically counting the number of mat offspring after going through one cycle. So it is a measure, it is really, how many mat babies do the derived types leave relative to the ancestors, and that involves a competition where they go through one life cycle.
Perhaps my question is, how do you ensure that that entity has formed at that point?
Has
Form.
form.
Yeah,
Form and what?
as in, now you can talk about a colony. So
Yeah.
my point being that maybe at that competition experiment, they only competed on the lower.
No, I mean, the form is given. Maybe form is, maybe another word you're thinking is boundary or discreteness, you thinking? But that's given by the walls of the tube. That is what maintains discreteness. And so we really don't, I mean, I could go through after the detail of how we do the experiment, but it is, the discreteness of the entities is guaranteed by the fact of the walls of the flask and the way we do the experiment. We really are measuring the reproductive success of the lineage. Of course, it's comprised of cells, but this lineage is recognizable, it's genotypically distinct, we can pick it out of the freezer. Okay.
Okay, so let's break for 20 minutes.
Thank you.
Okay. When you go from step 1 to 2, you get these multicellular things. In your case, you have this bottleneck.
Yeah.
But sometimes you have to get rid of this, and they have to reproduce as an entity. So they have to become a new entity held together to reproduce without the bottleneck.